Hello and welcome to Science Radio. In today's segment, we continue with our coverage of the workshop on next generation sequencing held at IISER Pune from the 24th to the 27th of February this year. In the previous episode, we heard from the coordinators of the workshop, Professor Sanjeev Galande.
So in that sense, it's a technological leap.
And Dr. Farhat Habib.
A single operator could generate millions of sequences.
We also heard about the talks presented by Dr. Vineet Sharma.
Recent studies have shown that 98% bacteria cannot be cultured.
Dr. K. Thangraj.
In India, we have large number of population groups. If you take any other country, any continent, you don't have such a huge amount of human population diversity.
Dr. Mithali Mukherjee.
NGS is like a tool. People should not get into that because I have the tool, I can just about answer or get the answers for everything.
Dr. Krishnapal.
By studying the chromatin states, we can see the transcription. We can decode actually the transcript level.
And Dr. Amit Dutt.
One of the problems that normally in the cancer field that happens is that a cancer as a disease is a very complex disease.
We also heard how next generation sequencing has made an impact on their research.
Most of the discoveries which is happening in my laboratory is NGS based.
Cheaper and higher throughput is what we want. And if you don't have that, we cannot do. So only next-gen sequencing.
The next 5 years, we will be using it very extensively. So
we need to generate our own data. So in that sense, it's very, very important.
If we implement all these NGS technologies which are recently developed, eventually the information we'll get can be directly translated to the application.
This is Science Radio on Vidyavani 107.4.
I found the workshop to be interactive.
it's the right time to have these kinds of workshops in different parts of the country because
Such kind of workshop will help students and colleagues to use more and more NGS data.
It's absolutely essential because
This is creating awareness as well as it is also bringing in more number of people who know what things are.
In today's segment, we will find out from the speakers about the importance of workshops on next generation sequencing.
I found the workshop to be interactive. I'm coming from a very specialized field, and usually in these workshops, which is very much required in these days because of the newer technology that is coming in, I had a feeling that the students who are coming from very plural background, primarily people from microbial and plant, where majority of the laboratories here, and so I was expecting not much of an enthusiastic response onto a cancer talk. But I was pleasantly surprised that I was interrupted during the talks. And over the coffee break that followed up, a lot of students approached up to me and they introduced themselves and they had like their own interesting ideas. So I was pretty impressed. So this is a great primer. These kind of workshops is a great primer for them to a technology. And what I presented today to them is that how this technology could be used in certain disease settings. Unless you really play your card, unless you really attend to these workshops, you really don't get to know what are the insights. It was well received among them and I could see that the students were pretty enthusiastic for this technology in general and then to the application of cancer as well. So I think the way it is being organized, and special thanks goes to Dr. Farhat Habib for organizing it.
The goal of this workshop was to give basically knowledge of NGS to the people or to the scientists who are working with the wet lab part of the biology, and most of the tools we introduced are basically user friendly because one of the problems with the NGS technologies is the amount of data you get, and most of the biologists are unaware of handling such huge amount of data. And so one of the goals was to give a way to handle this data and interpret it. So we hope that such kind of workshop will help students and colleagues to use more and more NGS data.
it's the right time to have these kinds of workshops in different parts of the country because people are clueless. Earlier times, if people would walk into the interview, we would ask them what you want to do. They would say biotechnology, right? And then they would say immunology. So they knew the discipline or they knew a buzzword or molecular biology. But what you can do with that is what the students need to appreciate. And since this is not taught formally in any of these MSc courses, hardly I think there are any universities which offer genomics as a course. So when the students come to us, we have to actually prime them from the beginning. And if they have orientation to these workshops, they have a research problem, then they can think where they can apply it because these are really hands-on experiments. I think it's important. For me, otherwise, the students keep on telling me, we'll do NGS. But why you want to do NGS? Because it's a fancy thing and it's the next buzzword. So they have to understand that even if they take an NGS problem, if they do a experiment today, if they don't know how to do the analysis 2 years later also, they will still be groping with how to do the analysis. So that's where I think it's important.
It's absolutely essential because India has so many research institutes, so many universities and so many colleges. But if you take the institute which has the facility to have such kind of workshop to generate knowledge and to give knowledge to the students or the faculties are very limited. IISER is one of those institutes have all the facilities and also the mandate to teach people, to teach the faculties who can understand A to Z of every technology we talk about. Therefore, I really appreciate IISER has taken up such kind of initiatives to teach or have such kind of courses to generate and spread the knowledge to the rest of the student and faculty communities.
This is creating awareness as well as it is also bringing in more number of people who know what things are, what softwares are available. Now they have a background of the different areas in which NGS can be used, like transcriptomics, ChIP-seq, or metagenomics, or human genome analysis, or different eukaryotes and plants. It is very important to train more and more number of people who can, contribute and work on these kind of projects.
We will also hear firsthand from some of the participants at the workshop about their expectations and experiences.
By and large, as I see, the workshop is really going very well. It is very informative. They've given us a detailed information on everything that we need to know for the bioinformatics analysis.
This was a completely new experience for me, and basically my aim of attending this was only to get the concepts right, like the basics right? And the tool that has been introduced over here is from my department itself, and I'm going to be working on that.
I like the IISER Pune first of all, and then it's for beginners. So we don't know anything about this Unix commands. So they're telling from very basics. So now in 3 days we are very much familiar with that.
So I'm looking at next generation sequencing, application of next generation sequencing in clinical grade genomics. In the Western world, this technology is matured where they have applied essentially next generation sequencing to identify genetic mutations in tumor samples, and based on that they are giving therapy. So it's part of the whole personalized medicine paradigm. So I'm here to really understand the computational details of how to deal with the data.
They should explore the new areas of sciences for their future careers.
And that's where I think the NGS technology is important, and I would suggest that we should adapt to it and use more and more.
One has to have collaboration with many institutes working together in the same field.
If you want to go to genomics, you have to go with a working hypothesis.
Whether using NGS or whether without using NGS, go after the biology, less after the technology.
This is Science Radio on Vidyavani 107.4. The speakers at the workshop shared some words of wisdom for our listeners.
What we see is that during the PhD interviews, the students generally choose the areas which they are comfortable with, like molecular biology, immunology, which they have learned in their MSc, and generally they don't go for the newer areas like neuroinformatics, metagenomics, next-gen sequencing, bioinformatics. They should explore the new areas of sciences for their future careers. We will also have more experienced hands to do those things then.
The area of genomics, actually it's a little complicated. The Indian genome is totally different compared to the rest of the genome. If you want to actually make impact of understanding the Indian genome, then we need to go for very extensive analysis, large number of genotyping, then go for large number of genome. Whatever you take, one has to have a complete genome sequencing. So what is my suggestion for those who are in the beginning stage, at least to analyze the genomic aspects, we need to take up in a large scale, covering large number of populations. If not individually, at least one has to have collaboration with many institutes working together in the same field, try to generate huge amount of data to interpret in a better way.
People think that genomics is all about data crunching. My colleagues who have been trained in cell biology, they think that when we do genomics, it averages out everything. The other school of thought is that if you're working in a single cell, then it approximates for so much of variability which we know is in the genome. You cannot actually predict the outcome would be the same if you go to single cell. So each of them has its merits. You should use genomics for finding out broader answers. But I would say that if you want to go to genomics, you have to go with a working hypothesis. Otherwise, you just can be lost.
I think NGS has a huge potential. Earlier, most of the scientists were targeting individual genes and some individual pathway, but nothing is working in isolation. So it's a system which is reacting to any drug or any medicine or any perturbance you are doing to it. We need to think more and more globally. And that's where I think the NGS technology is important, and I would suggest that we should adapt to it and use more and more.
Don't go after the buzzwords. It's not the NGS that should allow you to make any decision at all, whether you should be getting into or asking questions in cancer. These are just tools. It could be microarray, it could be different technology, and today it is NGS. And these technologies are so much at ferment, we still don't know what path it is going to go tomorrow. What should be most important for you? Your question should be centered to the biology. You should be interested in the cancer basic biology and trying and understanding the complexities of it, so that could help you understand the underlying biology. And while doing so, you can use n number of different technologies, of which NGS could be one of it. So have less and less emphasis on the technology and more emphasis on the underlying biology and understanding the biology. That could be by using model organisms, or that could be using vertebrate system now that it is so much amenable to do so. Either ways, it is fine. So if you do not have a basic centric question that allows you to drive, the technology would be of no good use. I know people like taking tumor samples and doing the sequencing, but because there was no centric main question that was driving it, these technologies also have a lot of pitfalls. It has a lot of noise. So you need to have an hypothesis before using these tools in hand. So my advice to the student is, go deeper into the biology, make your own hypothesis, and try and do experiments to falsify it. Show that this hypothesis, what you have made, is false. And while doing so, in the rigorousness of that process, you would be able to say that, no, I mean, with all the set of experiments that I have done, whether using NGS or whether without using NGS, I am not able to falsify the hypothesis that I had initially made. Go after the biology, less after the technology.
With this, we come to the end of today's episode and the series covering the workshop on next generation sequencing held at IISER Pune from the 24th to the 27th of February this year. But before we conclude, let's hear some comments from our coordinators, Professor Sanjeev Galande and Dr. Farhad Habib.
This was our conscious decision to learn everything in-house, both the chemistry part as well as the analysis part. So one is a wet bench part of it, and other is a dry lab part of it, where you just sit on computers and analyze data. We realized that if we want to make it big in this field, we have to learn all of this and not outsource these parts. We then realized that it's the analysis that is more important. When it comes to analysis, nobody can understand that better than you yourself, because you know exactly what you are trying to answer. You know exactly what you're trying to look for. And that is the reason why we spent some time, invested a lot of money and effort in trying to learn these.
Well, one thing I would like to add for wet lab biologists is to learn a little bit of programming or scripting in their repertoire. Very quickly, you'll find that you're dealing with data sets, even if they're not large in the sense of, say, Facebook or Google scale large. Even if you're dealing with a few thousand lines of data, it becomes quickly unmanageable unless you know formal ways of handling it. And sometimes you can get by with, say, trying to do things in Excel and so on, but it's very, very easy to make costly mistakes there.
So for us also, I think it was quite an eye-opener. We had our own expectations from this workshop. We had also projected that how we want to do this. But of course, since this was the first time, we have learned our own lessons. Of course, most of these are good lessons that we would like to improve, so that next year we are hoping to have this again. And at that time, we'll incorporate all these things that we have learned in this first experience. And from the feedback of the participants, which has been mostly quite positive, they were all happy and they thought that now they got the confidence. That's, I think, one of the most important achievements of this workshop.
Other than that, I would like to say that I was thankful for all the support that was extended by the Center of Excellence, especially Sanjeev and IISER Pune, Professor Shashidhara helped us a lot with making sure that things went smoothly, and it wouldn't have been possible without the support from other staff in IISER, especially the IT team, which made sure that the computer room was working a day before we started our workshop. Without that, the hands-on side would simply have not been possible. And that was one thing that was really appreciated by a lot of people, that there are a lot of workshops across India where people give you lectures and you try and figure out what you can from them. But without the hands-on component, you really don't get a feel of what it actually involves.
Do write to us with your comments and feedback. Our email ID is radioiserpune@gmail.com. That is radio, IISER Pune@gmail.com. This is your host, Sourajit Basu, signing off.